Hello, and welcome to my presentation on cloning animals by somatic cell nuclear transfer. I find it to be a very interesting topic that's surprisingly easy to understand and has a lot of elements of cell biology to be considered about it. So let's get started. My query question for you is: explain the general mechanism by which somatic cell nuclear transfer works for cloning animals. Explain key factors that can limit the success of somatic cell nuclear transfer and why research is still being done to overcome these limitations. Summarize the ways in which epigenetic reprogramming plays a role in the ability of an embryo to develop from the genome of an adult animal, and some of the ways this is similar to what happens during the normal fertilization of an egg cell. What is one way cell signaling must be considered for creating embryos this way? Compare and contrast the factors that affect the efficiency of somatic cell nuclear transfer using cell nuclei from donor cells in different stages of the cell cycle. We'll go over the differences between using the nuclei from G0 cells to M-phase cells. My student learning outcomes for you are: by the end of this presentation, you should be able to describe how somatic cell nuclear transfer works for creating animals that share the same genome, some of the ways epigenetic reprogramming plays a role in the success efficiency of somatic cell nuclear transfer, at least one distinct element of cell signaling present in normal activation of embryonic development that must be artificially simulated in creating embryos with the genetic material of adult cells, and some general reasons that it matters which stage of the cell cycle the donor cell is in at the time its nucleus is removed for transplantation into the egg cell. So, somatic cell nuclear transfer, abbreviated SCNT, also just called cloning or nuclear transfer, NT. It is a process by which the cell nuclei of adult somatic cells from an animal that you are trying to clone, such as this adult dog here, into Snuppy, who was the first clone dog, with his surrogate mother here, who the egg cell that had its nucleus removed and implanted with the adult egg cell, or the adult cell nuclei from this dog here, was put in the uterus of this dog, and out came Snuppy here. As the cell nuclei of the adult somatic cell contains the genetic material that is the same as this dog, and it developed into this one. It has a very low success rate based on the number of attempts to recreate embryos using the genetic material of adult somatic cells in comparison to the number of clone animals that are actually born from these attempts. About 95 to 99% of these attempts fail in general. This is largely attributed to unsuccessful epigenetic reprogramming of the adult somatic cell genome by the enucleated egg cell to produce totipotent embryonic stem cells. Stem cells that can differentiate into all of the kinds of cells in the body, including embryonic cells. Other factors can play a role as well. Scientists are using cell biology to figure out ways to improve this efficiency, and I included this little fun fact because it kind of helps me to think about this conceptually. The doctors were convinced that I actually had a twin in the womb who didn't make it past a couple months. And, you know, if you were to imagine hypothetically I were the first clone human and there were about 100 enucleated human egg cells implanted with the old Mies somatic cell nuclei, about 95% of them would have died immediately or failed to have been activated by cell signaling, which we'll talk about. And 2 would have taken as an embryo, and I'd have been the one that made it, and that embryo of my twin clearly wouldn't have had successful epigenetic reprogramming so that all of development could take place from totipotent embryonic stem cells. And here are some important points about that epigenetic reprogramming. After the adult somatic cell nuclei is implanted into the enucleated egg cell, the egg cell that's had its nucleus removed, the histones that package the chromatin in that nucleus are replaced with histone variants that are stored in the egg cell and imported into the nucleus. These histones also have to go through a lot of changes to their histone modifications. Histone demethylase enzymes have been found to be important in certain cases for successful cloning. And improved cloning success has been observed with the use of histone deacetylase inhibitors. So that's small molecules that inhibit histone deacetylases and thus leave more histones acetylated and more open chromatin for gene expression, a lot of gene expression and development. And in normal development, DNA must go through extensive demethylation to reach minimum methylation in an embryonic state, while stem cell while somatic cell DNA is heavily methylated. And this is a major area of further research on improving SCNT efficiency because it's elusive how much this matters in every case, and also what exactly goes on and what has to go on for completely successful cloning. Animals often get health problems more when they are cloned than when they are original. And that's thought to be because of unsuccessful epigenetic reprogramming. Normally, in fertilization, phospholipase C zeta activates a cell signaling pathway where PIP2 is cleaved to diacylglycerol and IP3, which acts on the IP3 receptor in the endoplasmic reticulum, which causes the release of calcium ions and oscillation. And it also leads to the degradation of maturation promoting factor, which allows the artificially fertilized egg cells to exit M phase and initiate development as blastocysts and divide on to create embryos that eventually develop into the full-grown animal. But since this phospholipase C zeta is not present in the cell nuclei of somatic cells, these conditions have to be artificially simulated, and a number of agents are used to simulate this process depending on the species. And other conditions such as coordinated phases of the cell cycle for the donor cells and egg cells, because presence of things like MPF matters in what needs to be done to activate these early embryos. And other factors involving cell signaling that seem to play a role involve transcription factors, which is still being investigated. Key points about the efficiency of cloning using cell nuclei from donor cells in each phase of the cell cycle. As you can see in this diagram here, the nuclei in S phase and G2 phase have double the amount of gene material present in G0, which would have been here, and G1. And the normal amount of genetic material in fertilized eggs is compatible with the amount of genetic material in G1, making G0 and G1 phase the best phases to take cell nuclei from in general, just because they have the right amount of genetic material. And there is some variability between these two depending on the cell type that it's taken from. A study found that cattle were more successfully cloned from the nuclei of non-transgenic fibroblast in G0 than the nuclei of non-transgenic fibroblasts in G1, different cell types. Well, these cattle were more successfully cloned from the cell nuclei of transgenic fibroblast in G1 than transgenic fibroblasts in G0, showing that the efficiency of cloning by somatic cell nuclear transfer with donor cells in either of these phases depends on the type of donor somatic cell type. This cell type was better in this phase, and this cell type was better in this phase. Then, of course, in S phase and G2 phase, it's much trickier, has been done, but is much trickier because you've got partially replicated chromosomes in S phase, and that leads to the incorrect ploidy for a typical fertilized egg cell. But if the phases of the cell cycle are coordinated between those cell types, you can get it so that it just divides and takes both copies with it into different cells. So it can be done, but it's harder and, more complicated. Deals with N phase, having the nucleus break down and chromosomes wherever they are each phase, but it has been done. Some comprehension questions. Why is cloning from adult cells that are in S phase considerably more difficult than cloning from adult cells that are in G0 or G1? I'll give you a minute to think about all of these, and then the answers are on the next page. What happens to the chromatin in the nucleus from the donor cell? What happens to the chromatin in the nucleus from the donor cell after it is transplanted into the nucleated egg cell in a successful cloning? And finally, what happens if the effects of PLC zeta in normal development cannot be recreated for the enucleated egg cell artificially implanted with the donor cell nucleus? And here are the answers. In S phase, the genome is partially replicated, resulting in excess alleles of some genes. So the nuclei of these cells do not have the correct ploidy of genes. And here, what happens to the chromatin in the nucleus from the donor cell after it is transplanted into the enucleated egg cell in a successful cloning? First, it has many of the histones comprising its nucleosome core particles switched out for histone variants stored in the egg cell and imported into the nucleus as they would be. And its histone modifications are changed to optimize gene expression for development. Then what happens if the effects of PLC zeta in normal development cannot be recreated for the enucleated egg cell artificially implanted with a donor cell nucleus? The fertilized egg cannot exit M phase and develop into a blastocyst to become an embryo. Here are my works cited. I got a few things from sources other than the textbook. One of those figures here, a picture of Snuppy with the dog he was cloned from and his surrogate mother. And then there were a couple of studies on somatic cell nuclear transfer involving the cell cycle and epigenetics that I read about, and you can read about here. They're quite impressive and intriguing. Thank you.